Hello, everyone. My name is Apoorva Chowdhary. I am a second-year medical student at UW Spokane, and my poster is on improving diagnoses of ocular surface diseases. So let's start with the background here. So ocular surface disease is a term that encompasses serious and vision-threatening conditions caused by a pathogenic microbial organism, ranging from bacteria to parasites. And there is a lot that's unknown about the microbiome of the eye, but its complexity often makes these infections really difficult to diagnose. And culture is currently the gold standard for diagnosis of these conditions, but corneal cultures have been shown to only have 60% sensitivity and take days to yield results. So in this study, we wanted to investigate the utility of a new device in efficient and accurate diagnoses of ocular surface disease, the Oxford Mini-Ion Nanopore. So this is a small USB-sized device that can sequence DNA samples from multiple patients at once in a wide variety of settings. And it has already shown promise in diagnoses of various conditions, including Ebola, lower respiratory tract infection, and endophthalmitis. So our main objective was to develop a time-efficient, accurate, and widely applicable diagnostic protocol for ocular surface disease using this device. So now let's move on to the methods. So we purposely kept the inclusion criteria wide. Excuse me. Try to do this again. There we go. So we kept it broad for this study so that we could investigate the applicability of our protocol with the diverse set of patients suffering from ocular surface disease. And these patients were recruited by referral from clinicians, and the diagnoses of the disease, recruitment, ophthalmic exam, culture, and swabbing were performed on the same day. So the graphic on the right here kind of shows our process. So we would collect the sample, store it in Zymogen DNA and RNA shield just to maintain the integrity of the DNA, and get culture at the same time as our control. We would then extract genomic DNA using the Quiagen DNeasy blood and tissue kit. So these samples were transported to the lab. Extraction was performed and amplification was performed using the Repligy ultrafast mini kit. And at both of these steps, we measured the concentration of the DNA. And then library preparation using Oxford Nanopore's rapid barcoding kit was performed as well. And all these steps were relatively quick, so this would take maximum 2 to 3 hours in the lab. And once the Oxford nanopore flow cell, which contains little nanopores that sequence the DNA, was prepared, we would perform sequencing using the Oxford Minnow software, which actually gives you sequencing reads live. And this was really helpful for us so we could see if our sequencing results were failing right away and then terminate the sequencing. But we actually developed a smart tool in-house, and this has been in place for a while. So we would use this to align sequences that we were sequencing to a database containing sequences from the NCBI taxonomy database. And so the taxonomy database includes like bacterial, fungal, parasitic DNA sequences, and significantly elevated species level counts relative to the expected background from the SMART database were considered positive hits. So an organism was considered diagnostic if it represented the most abundant reads in the sample, and then these results were to be compared to results from the culture. So as you can see on the left here, an important point was that the Minnow software requires a minimum of 10 microliters of DNA at 40 nanograms per microliter for long read and high throughput sequencing results. And unfortunately, as you can see from our table here, we struggled a lot with DNA yield, and we made multiple adjustments to our methods to account for this. We changed the swab from alginate to rayon, as studies had shown that rayon had better DNA yield. We actually implemented the Zymogen DNA and RNA shield early on in the study. So all of these samples were collected using that method, but beforehand we weren't doing that. And we even had lower DNA yields than this, next to nothing. But either way, we struggled a lot, and despite the adjustments, we weren't able to get DNA concentrations that yielded good sequencing results. So where do we go from this? Oh, well, let's talk about it. So there are additional modifications that we could make to our sequencing protocol or beforehand to optimize DNA yield that we're looking forward to doing in the future. So these include recent developments from Oxford Nanopore that would allow us to make changes on the sequencing end to enhance the amount of microbial DNA versus host DNA, which we're sequencing. And this is referred to as adaptive sequencing. So the device can be programmed so that when non-microbial sequence is read, the bias voltage of the nanopore is switched so that a DNA strand, instead of being sequenced, is actually ejected from the nanopore, which would reduce the amount of host or human DNA that we're sequencing relative to microbial DNA. There's also an additional enrichment step that can be formed prior to performed prior to sequencing, involving using protein A paramagnetic beads that bind to methylated host DNA to deplete the human DNA from mixed DNA samples, once again giving us more microbial DNA. And as you can see, low sample size was also a limitation within our study. So we did conclude that although we developed a foundational diagnostic protocol for ocular surface disease using the Oxford Nanopore mini-ion device, further studies utilizing methods to optimize microbial DNA yield are necessary to determine the accuracy and the applicability of this protocol. Thank you.